Thank you all for joining us this afternoon and taking some time from your busy schedules to learn a bit about the research that's going on here at the University of Georgia as it relates to the SARS-CoV-2 virus and the COVID-19 disease. This is going to be the first in a series of 3 Fireside Chats, and my name is Michelle Bowden, and I'm going to be your moderator for these next 3 chats. I am the Director of Business Development here at the College of Veterinary Medicine at the university, and I'm joined by 3 colleagues that have backgrounds in infectious disease transmission and diagnostics, and have moved over into the SARS-CoV-2 space to try and understand and help disseminate knowledge. And there are Doctors Mark Jackwood, Jesse Hostetter, Fred Quinn, and Chet Joyner. And before we get started, I just thought I would give people a little bit of background about the importance of academic science. You typically see these type of pyramids in drug discovery, pharma, business development, and the base of the knowledge that biotech and pharma draw on to develop vaccines and diagnostics and therapeutics that you take on a regular basis, that foundation is set by your academic scientists. They are the key opinion leaders. They are the experts in the field. They identify targets, they identify mechanisms, and they disseminate this into the scientific public, and biotech and pharma draw on this. So it's extremely important what our academic scientists are doing and what they're contributing to this knowledge base. And I hope you agree as we move forward in these talks, the vital nature of the work that they're doing. So with that, I will set the scene for everyone. We're first going to start off with a history of coronaviruses and where did they come from. And that discussion is going to be led by Mark Jackwood. So Mark Jackwood is the J.R. Gleason Professor of Avian Medicine. He heads the Department of Population Health at the college, and he himself has been working in obscurity for decades on coronaviruses, in particular viruses that infect poultry. So he studies genetic diversity of these viruses, mutation rates, kind of the evolutionary trends to understand how they emerge and how they move from species to species. His lab in particular is developing molecular diagnostics, and he's also developing reagents that are causing the backlog in the ability to test for this particular virus throughout the country. And so I'll let Mark go ahead and give you some background on these viruses. I will also say before we get started, please feel free to ask questions in the Q&A section, and we will do our best to bring those questions to the attention of the speakers in the various sections, and whatever questions we don't get to, we will do our best to address them at the end. We will also do our best to minimize the scientific jargon and the acronyms so that you all have a better understanding of what we're actually talking about when we're not talking in code. So with that, please meet Dr. Jackwood.
Thank you, Michelle, and good afternoon, everybody. So let's go ahead and jump right in. And one of the first questions that people start asking when we have an outbreak of disease like this is, where did this virus come from? And for coronaviruses, we know that coronaviruses that infect and cause disease in mammals, including humans, that the reservoir for those viruses resides in bats. So the question is, why bats? Well, there's many, many different species of bats, over 1,200 species of bats. So this provides a very diverse population of animals to be able to harbor all these different coronavirus types. And there's, thousands of coronaviruses that have been identified in bats that are unique. The bats seem to tolerate the viruses very well, as a reservoir definition goes, that the virus can be maintained in that species without causing severe disease or illness. But the diversity of those viruses in bats allows them, when they have the opportunity, to jump to other species sometimes. Now, typically coronaviruses are very species specific, but when the opportunity arises, they can jump. And this happened with SARS coronavirus and the MERS, or the Middle Eastern respiratory coronavirus, that we've seen some years ago. What happened with those two is they jumped to an intermediate host first. The intermediate host for SARS was the raccoon dog and the palm civet. The intermediate host for MERS was the camel. Once they got into those intermediate hosts, they then were in close contact with humans. The virus changed ever so slightly to allow it to then jump to humans and then be transmitted among humans. So for COVID-19, or the SARS-CoV-2 virus, there has been an intermediate host that's been speculated to be the pangolin. I think the jury's still out on that one. I don't think that we have quite enough scientific evidence to show that that truly is an intermediate host for the SARS-CoV-2 virus. But there is a bat virus, a horseshoe bat virus, which is named RATG13, which we've known for a little while, since 2013 actually. And that virus is extremely similar to the COVID-2 virus. It's entirely possible that this virus jumped directly from bats directly into humans. And I'll go through that in a minute, because the host receptors, the host cell receptors, are very similar. Now, a lot of people have been asking about birds and what role they play in this particular virus. And I think that question a lot of times comes from the role that birds play in flu, which is very different. Here, the coronaviruses that infect birds, including chickens, poultry, pet birds, they are not the same as the mammalian coronaviruses. And up until now, none of the avian coronaviruses have been shown to be able to infect mammals. Next slide, Michelle. So coronaviruses are divided into different groups based on their genetic and antigenic characteristics. And you'll see that we have, they're named alpha, beta, gamma, and delta virus groups. The alpha viruses are mammalian viruses. They include viruses like transmissible gastroenteritis virus in pigs, feline infectious peritonitis virus, canine coronavirus. There's a human coronavirus 229E, which is in that group. The beta coronaviruses are also mammalian coronaviruses, and they include the viruses like bovine coronavirus, as well as the human coronavirus OC43, as well as MERS, SARS, and the SARS-CoV-2, or the virus which is causing the pandemic now. The avian group, or the gamma coronaviruses, include infectious bronchitis virus and turkey coronavirus. And as I mentioned, these viruses do not infect mammals, only birds. And the delta coronaviruses are viruses that infect mainly songbirds. You also see that coronaviruses can cause quite a wide variety of diseases. This COVID-2 virus causes a respiratory disease like SARS did, like the other human coronaviruses cause. We also have the bronchitis virus in poultry is a respiratory disease. Bovine coronavirus can be respiratory. But some viruses cause enteric disease, like transmissible gastroenteritis in pigs, turkey coronavirus, and canine coronavirus. And then we have a couple of viruses that can cause hepatitis as well as neurologic disease. Next.
Mark, so there was a question that came in about whether or not we as humans might be able to infect some of these other mammals or birds. Is there any evidence for that?
So yes, there is. I don't know if that's on the next slide or not, but the COVID-2 virus has been shown to infect cats, and it's also been isolated from dogs. No, not this slide. It'll be the next one, but we'll get to that. So it can infect, COVID-2 can infect cats. It can infect ferrets, and dogs have also been isolated, the virus from them. The route is from humans to animals. There's no evidence, to my knowledge, that any animals, no cats or ferrets, have actually transmitted the virus back to humans. As far as our animals that are involved in animal agriculture and food, there's no evidence that COVID-2 virus infects them at all. One of the reasons for that is the genetic similarities among these viruses can be quite different. Coronaviruses are a very diverse population of viruses. And as I mentioned, the COVID-2 virus likely originated from a bat, and you'll see that RATG13 bat virus is 96% similar across the genome for the COVID-2 virus, indicating that they are very likely, they're closely related. The SARS-CoV-2 virus is only 79% similar to the original SARS virus and only 50% similar to the MERS, or the Middle Eastern Respiratory Coronavirus. Just for comparison, the avian viruses are only 54% similar to the SARS-CoV-2. So anything less than, the upper 90%, 95 and above, are viruses that are very, very different from each other, and it's very likely that they're not going to cross species. Next slide. So a little bit about the host cell receptors for this particular virus and why it is somewhat species specific. SARS-CoV-2 uses what's called the ACE2 receptor on the surface of host cells. That ACE2 receptor is also found on cells of bats. And so we see that the similarity between the receptors allows that virus to go from a bat jumping into a human. The same was with SARS coronavirus. The original SARS also used the ACE2 receptor. Now the MERS coronavirus uses a different receptor on the host cell. It's a dipeptidyl peptidase 4 receptor, but bats also have that receptor. So we have bats with a similar, host cell receptor that humans have, and it allows those viruses to jump. Transmissible gastroenteritis in pigs, for comparison purposes, uses an aminopeptidase N as its receptor, and infectious bronchitis in poultry uses sialic acid. So these viruses use very different receptors, and the receptors that we have as humans can be quite different from the receptors that the animal coronaviruses use. And the inset there shows that COVID-19 viruses can infect obviously humans, bats, and cats and ferrets at this point seem to be hosts for the virus. Next slide. So the other factor that's involved in whether this virus can infect a particular host or not is the spike protein on the surface of the virus. And this is a glycoprotein, which means that there's just sugars attached to the protein itself that gives it its structure. The spike protein is made up of two subunits. The outside portion is the S1 unit, and the part that anchors the spike to the viral membrane or the surface of the virus is the S2 portion. The outer portion, the S1 portion, has what we call epitopes, or areas on the protein that are used by the virus to attach to the host cell receptor. Those areas are also serotype specific, so they give the antigenicity of the virus its character, and those epitopes are what induce the antibodies that will neutralize the virus and protect the host. So, and this is why vaccines that we produce or we try to develop against coronaviruses are most always targeting the spike glycoprotein. Okay, next slide, Michelle. So I just want to reiterate this point is that livestock and poultry and their products are not considered a source for COVID-19 infection in humans. We know that the most likely way you're going to get infected is by the person standing next to you. Respiratory droplets, and Dr. Quinn's going to cover the transmission of this virus in just a minute, but those routes of infection are through your nose, your eyes, and your mouth. And this is why it's very important to wear the protective gear when you're out in public. Next. So what about vaccines? These viruses are so highly infectious that it's almost impossible to control them without vaccines. For coronaviruses, live vaccines make the most sense. And I'm a big believer in live vaccines because they induce a very strong local immune response, which is protective. The drawback of live vaccines, obviously, is their safety. They can heat back up again to cause disease. So whether a live vaccine would be approved or not is a very big question. Kill vaccines, they induce a strong systemic immune response. These are the antibodies circulating in your blood. And these antibodies can or may not be protective. It depends a lot on the strength of the immune response, the type of disease that's associated, and also the antigenicity of the vaccine. We do have vaccines against coronaviruses in animals. The vaccines are for canine coronavirus, bovine, just to name a few, feline, TGE and IBV vaccines. Almost all of these are live vaccines. Okay? And there are no, it says no vaccines for many animal coronaviruses because there's such a wide variety of diseases out there that it's almost nearly impossible to produce vaccines against all these different coronaviruses that we have circulating. The other point to make with this slide is that there's no approved treatments for coronaviruses in animals. And as far as I know at this point, none for humans either, although a lot of are being tested. So before we move on, a couple of questions that we have is this disease going to be seasonal? And you know, a lot of people speculate that it will be seasonal, and a lot of the experts in Washington are telling us that it's going to come back in the fall very strong. And that's very possible. I mean, it may circulate and be seasonal just like we see avian or like we see flu virus in humans. And will it be endemic? I suspect it likely will be endemic. We need to try to control this with social distancing for now, but in order to actually get a very strong immune response and get control on this virus, we're going to need vaccines to be able to do it. So hopefully we'll get some good vaccines coming out in the future. And with that, I'll turn it back to you, Michelle.
Michelle, I think you're muted.
Yes. Sorry about that. I'm a little Zoom deficient. So one question in your earlier slide, Mark, you pointed out that there were other human coronaviruses, in particular 229 and the OC43. And those viruses do seem to be endemic, and they also seem to be seasonal. And so I guess, would it be out of the question that once we're no longer naive to SARS-CoV-2, that these SARS-CoV-2 may become a seasonal cold virus, if you will? Or do we just not have enough information yet?
Yes and yes. So, you know, we can't predict the future. These viruses all behave very differently. It's very possible that when we do develop what's called herd immunity, and I think one of the speakers is going to talk about herd immunity here in a minute, that we can live with this virus and it would become fairly seasonal, like some of the common cold coronaviruses that we have. The virus can change. It can change. I've seen it go both ways. I've seen viruses change where they become more pathogenic and a much bigger problem than they are currently. And I've seen them change the other way where they seem to adapt to the host and cause just a very mild seasonal disease. I think we don't have enough information about this particular virus yet, but immunity is going to be a big factor in its control in the human population.
And so another question that's come in is, what would make the virus seasonal?
Well, if it's not going to be eradicated, and I very strongly believe it will not be eradicated, I think it's here to stay. We're going to have a portion of the population that's always going to be susceptible. These will be young folks that haven't been exposed to it yet. There'll be older folks that have a waning immune system that is not responding very well to pathogens anymore. And so we're always going to have that portion of the population that is going to be susceptible to these viruses, just like we do the common cold or influenza viruses. And the viruses can change too. So when the antigenicity changes a little bit, they can avoid the immune response to some extent. So that's where we'll see a seasonal event with this particular virus. That's really going to be complicated with flu, because flu, as you know, is also a seasonal event. So when someone comes up with a respiratory disease, we're going to need to sort it out. Is it coronavirus? Is it COVID-2, or is it influenza?
Great. And we will definitely be addressing the topic of vaccines, seasonality, changes in sequence of the virus in our chat number 2, which will be all about how our human immune system is reacting to seeing this virus for the first time. And then in chat number 3, where we're going to have an extensive discussion about vaccines and therapeutics and what we know and what we don't know in that arena. So now I think we will move on and talk a little bit about what we know about the transmission of SARS-CoV-2. And Dr. Fred Quinn is going to lead us in that discussion. He is the Athletic Association Professor of Infectious Diseases. He's also the Department Chair of Infectious Diseases at the University in the College of Veterinary Medicine. His background is quite extensive. Before coming to the university, he spent some time at the CDC and oversaw several emerging bacterial disease outbreaks around the country. He also has a current research focus in the disease progression and transmission of both human and bovine tuberculosis. And he is also involved in probably diagnostic testing, as well as contact tracing for SARS-CoV-2. And this is based on his background with tracing of tuberculosis. So I will let Fred jump into what we know about the transmission of SARS-CoV-2.
So thank you, Michelle. I appreciate it. And good afternoon, everybody. So today I would like to give you some information, some aspects behind disease transmission, some of the mechanisms. Because understanding disease transmission is a very important mechanism for ultimately allowing us to control a disease, particularly when a vaccine isn't available, which is what we're dealing with right now with COVID. And just as a way of background, there are several types of disease transmission, and you're aware of them. Foodborne disease transmission with pathogens like salmonella, bloodborne pathogens like Ebola and HIV, insect-borne pathogens like malaria and Zika virus, and the respiratory route that we're talking about here today for diseases such as tuberculosis, influenza, and now COVID. So I said today, for obvious reasons, we can focus on the transmission mechanisms, and there are several investigators at UGA that are looking at transmission processes for a variety of pathogens. And again, understanding these mechanisms will be very important. So starting with this slide, the first point, the respiratory disease transmission begins when a susceptible host is exposed to an infectious person. It seems like an obvious statement, but it is actually quite complicated. And as these two images show, a person who has a respiratory pathogen or a respiratory infection in his acute phase and is sneezing, coughing, speaking loudly, there's a lot of discussion in the news media about particle sizes associated with this COVID-19 transmission. You've heard about small particle sizes and large particle sizes and the pros and the cons and what that means. And hopefully this image is a really good characterization of what we're dealing with, because you can see there are small and large particles. And all respiratory pathogens deal with this. The smaller particles, obviously, they all contain potentially the pathogen, but the small particles having a better possibility of remaining airborne for extensive periods of time. Whereas the larger particles, sort of at the bottom of that image, you can see will fall fairly closely to the person who's generating the aerosol. There's, again, discussion about the large particles remaining on surfaces for extended periods of time, and we can discuss that, whereas the small particles remain airborne. And this is, again, the social distancing component that we can talk about, because these particles, like I said, can transmit pretty some distance. And in addition, this is a good indicator of why masks would be important, as well as social distancing, because obviously if this individual had been wearing some kind of cover, the amount of particles that would be dispersed would be significantly less. And the individual on the right, the diagram of the person inhaling or being exposed to these particles, also if he or she were wearing a mask, it would make a difference. And obviously, again, social distancing. So how is this organism spread? Hopefully, again, this is a respiratory route, but it is also the involvement of other mucous membranes. So obviously inhaling the organism, getting the organism in your nose, these are all mucous membranes, which is how the organism initially is deposited. But there are other mucous membranes. There's the eyes, and there's discussions obviously about if you touch a surface and rub your eyes, is that a mechanism for infection? It is. It's been demonstrated for a number of respiratory viruses, including influenza, able to access your bloodstream. What about other mucous membranes? There's even reports now sexually transmitted corona, but again, there's there's very little data on those other mucous membranes. Whereas again, the eyes, the nose, and the mouth, there is some pretty reasonable data regarding that. So coughing, sneezing, speaking loudly, you don't think about that quite as often, but that also can be a mechanism of dispersing particles. The particle mask question, someone did submit a question about the effectiveness of different kinds of masks. Obviously cloth masks are the most common. And again, if you look at the image on the left, a particle mask would be of significant help in preventing quite a few of those particles from escaping into the air. Would an N95 or a KN95 be more effective? Possibly. So the difference between an N95 and a KN95 is the source of the mask. So the N95 is a US designation and the KN95 is a Chinese designation. They're virtually identical in their construction and their blocking capability. The 95 in the N95 or N95 designation refers to blocking 95% of particles of a particular size, which in this case is 0.3 microns or larger, which is pretty tiny. But unfortunately, the COVID virus is between 0.06 and 0.14 microns, which means it's smaller than the pore size of these masks. So even though they will block more particles inward or outward, they're still not a complete shield. And so, again, looking at different kinds of masks and different kinds of options, there are improvements, and this would be an improvement, but it's not perfect. The other aspect of this is N95 masks require you to be fitted. You can't just go to a store and buy an N95 and make it perfectly effective. You were supposed to be fit tested for them. Individuals with beards like myself, you can't use them because there are gaps. And so anyway, there are pluses and minuses to all of these kinds of masks and face shields, but again, any kind of protection, again, looking at that image on the left would be useful. So the bottom point, risk of infection is dependent upon exposure time, dose, proximity, and host susceptibility are absolutely true. That's true of any infectious agent. And so if you are close to an individual for an extended period of time, you will receive a higher dose. And if you're a more susceptible individual, such as being over 60 years old, that makes a difference. So next slide.
Fred, can I ask a question before we move on to the next slide? So do we know how long people are infectious?
Oh, not really. And I can get to that a little bit more in a couple of slides from now when we're talking about, or actually it's the next slide with the carrier state and things of that nature. But as long as you have viable pathogen in your respiratory tract, you are able to transmit it. How long that goes for is debated. The current test for looking at it is the PCR test to see if you have virus in your nose, in your respiratory tract. The problem with that is it doesn't tell you if you have viable virus. It tells you have the nucleic, the RNA associated with it. It doesn't tell you exactly if the virus is still infectious or not. So that's a very complicated. It's a great question, but it's a very complicated one. And yeah, I think that we'll have a better handle on that, but the jury is still out, I think, on that. So that brings it to this slide. So this is just again an overview of infectious disease transmission with an emphasis on the SARS-CoV-2. So all of these interlinking points are important in the process of disease transmission, but let's look at these as they focus on the SARS virus. So the agent SARS-CoV-2 has all of these pathogens, or a pathogen would have a primary susceptible host and a reservoir. The reservoir means where is the pathogen residing to be able to be infectious. It may have other locations other than, in this case, the human host. And Dr. Jackwood did a really nice job of saying that this probably isn't the case, but again, we certainly know that humans are a reservoir. So this also brings up the question about primary susceptibility and less susceptible hosts. The primary susceptible human hosts, as we've seen very well identified, are the elderly, heart and lung issues, immune compromised, obesity, diabetes, liver and kidney disease, and probably others, where you are still able to be infected, but these are the individuals who will develop the most robust disease. On the other side, you have the reservoir, as I mentioned, which would be, in this case, the human respiratory tract. But you also have to consider the fact, and this has been discussed quite a bit in the media, about what's referred to as the asymptomatic carrier or the asymptomatic transmitter. So we know that individuals, like we showed in that previous slide, who are coughing, sneezing, and speaking loudly, who have the virus, who definitely have the virus in their airways, are transmitting, and that's again relatively easy to identify. But there's also, and this is true of most respiratory pathogens that have been studied, there is a group called carriers, and these are individuals who are completely asymptomatic, or for the most part, maybe a very, very small amount of symptomology. these individuals can have the virus in their upper airways, in their nose, and there's no immune response. You're completely showing no signs and symptoms, but you are able to shed and transmit. And when I teach the class, I use the example of Typhoid Mary. Obviously, this is a bacterial disease, but Typhoid Mary had absolutely no symptomology, but she transmitted the disease to about, at least known, about 80 people. So this is a very important point. It's very difficult to identify these carriers at this point, and it has a big impact on how we develop statistics, looking at transmission, et cetera. And we'll get to that on the last slide. The portal of entry, whoops, I'm sorry. Go back, Michelle, sorry. So the portal of entry are the mucous membranes, as I mentioned in the last slide. And then the means of transmission are aerosol, and also so there's direct and indirect transmission. A direct route would be what we're talking about here through aerosol. An indirect route would be something, an insect could be a vector, but in this case, indirect, we're talking about touching a contaminated surface and then touching your eye. So this pathogen can be transmitted by both means. Next slide. So there's a lot of terminology being put out into the media, and this is one aspect or one bit of terminology to clarify, and it looks at the how contagious a pathogen is. And so all infectious agents are given what is called an R0 value by medical personnel and epidemiologists, looking at how effectively an organism can transmit and how effective and thus be how contagious it can be. Without going into the mathematics, what this number refers to, and you'll see like measles at the very top says 12 to 18. What that refers to is if you put an individual in a room with 100 people, and the individual has acute transmittable disease, how many individuals can become infected from that single individual? And in the case of measles, which is the most infectious pathogen today on the planet, you can transmit, one person can transmit to 12 or 18 others. If you work your way down to COVID-19, it says 1.5 to 2.5. That is the current estimate. That number is going to change upward or downward depending on how our data are developed. Oops. How our epidemiological data are collected, how effective our diagnostic tests, and how more specific our diagnostic tests are going to become. But right now, this number refers to if an individual is in a room between, say, 2 and 3 individuals can become infected from this individual. And like I said, this number is going to change more than likely. It could go down as we find less and less people who are carrying the organism actually become infected. It could go up as we find our diagnostics get better and we find that individuals who have different kinds of symptoms have actually become infected. So we'll just have to see how this goes. But this is a starting point of where in the chart COVID-19 fits in. But again, a way we will ultimately control this, having this number, is with something called herd immunity. And next slide. So what is herd immunity? So herd immunity is a term that is again used by epidemiologists to explain or to demonstrate how an infectious agent is ultimately controlled within a population. So if you look at the upper left-hand figure, an individual who is again acutely infected and transmissible is in red, and the little blue people around him are individuals who are completely asymptomatic and have no protective capability. So they've not been vaccinated, they're not wearing they're not social distancing, they're not wearing face shields, etc. So how many individuals can become infected from this one individual? And again, the R naught data would say 2 to 3. But then you have to realize that within those 2 to 3 people, some of them will develop acute disease. They can then infect 2 or 3 others. They can infect 2 or 3 others. And this can continue on, if you'll move to the left side of these images, until you finally reach what's called herd immunity. And that's the point at which the population has now has enough infected individuals who have developed an immune response, a protective immune response, to where the transmission of the infection is now stopped. The estimate right now is that it will require about 70% of our population to be immune to this virus. And that is a very large number. And again, this is without a vaccine, because a vaccine would tremendously help develop herd immunity, again, as we show with measles vaccination and smallpox vaccinations years ago. That develops our herd immunity tremendously and doesn't put the population at fatal risk in another way of developing it. So this increase in herd immunity, one way or another, we will have to attain it. It's also possible that as our diagnostics improve, we will find a significant number of individuals who are asymptomatic but have been protected. Unfortunately, at this point, and I know Jesse and Chet will talk about this a little bit later, but we don't understand, we don't have a good enough background knowledge to be able to look at antibody response, for example, and know who is protected and who isn't. So this information will have to be gathered as we move forward, and ultimately we'll be able to understand or define when we reach the correct amount of herd immunity. So I'm sorry.
Fred, can I ask a question? So there was some talk yesterday by the governor that there's a monitoring tool for contact tracing, and that they're looking into adapting this tool along with the diagnostic test. So I'll do a time check. We're at 1:42 p.m. After this question, we'll move to the diagnostic, but maybe you could take a minute or two and address this app and how it might work.
Sure. And
your involvement in it.
So, well, I'm personally not involved in this. We have a similar study. Dr. Chris Whalen in the School of Public Health has been working in looking at tuberculosis transmission monitoring for many years, and has developed a similar kind of technology to monitor tuberculosis transmission within a population. But the basic concept, again, if looking at this herd immunity image, if the person in red contacts 3 people, you would like to know who those 3 people are. So the Public Health Service can then go and interview those individuals, monitor their health and well-being, see who they in turn have also come into contact with if they start developing some kind of symptomology or their test turns positive. So what this would do is provide the public health agencies with a significant forward leap. You're ahead of the transmission curve. You're not waiting for someone to come to a hospital with a disease. You're actually out there monitoring who potentially could develop it and putting protective measures in place to slow transmission. So this is just another mechanism, a very important mechanism. If it can be acquired, it's a lot of work and it takes a lot of resources. And the phone app is extremely helpful in monitoring and being able to stay ahead of the curve.
Great, thank you, Fred. So now we're going to move on to some discussions around the different types of diagnostic tests that are available, what these tests tell you, what they don't tell you. Dr. Jesse Hostetter will talk first about the molecular tests, which were the first tests that were rolled out. Jesse is the Barry G. Harmon Professor of Pathology at the College of Veterinary Medicine. He is a trained veterinary pathologist, and he also heads up or is the executive director of the Georgia Diagnostic Laboratories. His main area of research has been infectious diseases of livestock, mainly cattle. And he and his group at the Georgia Diagnostic Laboratories are ramping up to get what's called CLIA certification in order to start testing and take the burden off of the lack of testing in southern Georgia. And CLIA stands for Clinical Improvement Laboratory Amendment. It's one of the two approval mechanisms for diagnostic tests. The other is through the Food and Drug Administration. So, Jesse, I will let you run with the molecular diagnostics.
All right, thank you. So I guess so that we're all on the same page, there's really two kinds of diagnostic assays that are available, and one will detect the virus for SARS-CoV-2, and the other will detect an immune response, and this is called serology. And what I'm going to focus on is tests for the virus itself, and then Chet's going to talk a little bit more about serological tests. So when we talk about detection of the virus, there's a test that's available, and you may have heard this. It's called the viral test or the molecular test. And what this does is it tests or detects a component of the virus, and that's genetic material. And so we're looking for RNA that's specific for SARS-CoV-2. Right now we have one test that's approved. This is used on swabs, and it's really kind of the gold standard that we have right now. Another has been approved that can be used with saliva, and we're waiting for that to get ramped up. I'll tell you that there are a lot of tests and test platforms that exist out there. The more that we can use, the faster and more capacity we have for testing. And I'll tell you that Dr. Jackwood, who you heard from earlier in the College of Veterinary Medicine at UGA, is working on validating some of these tests and reagents to increase our toolbox for tests that we can use to ramp up our capacity. And so we have these molecular tests, and they use a few different types of samples. And by far and away, the most common is the nasopharyngeal swab. There's a picture of that on the slide, and I think many of you have heard of this. So the swab is placed in your nose, and it travels back through your nasal cavity all the way back to the very back of your throat. And as you may imagine, that is not the most comfortable thing in the world. Some of you may have had that done already. That samples the upper respiratory tract, so the nasal cavity and the throat. There's also you can collect sputum, and that's coughed up material, and sputum will test the lower respiratory tract, which is the lungs. As I mentioned, there's a test now that's been approved for saliva, and I'll talk a little bit more about that in just a couple of moments. The important thing to keep in mind with these tests, the viral test, the molecular test, is we have to have sufficient sample. And so we really need at least 10 viral genome copies in order for the test to detect infection. And a question that I know everyone has is really who should be tested. And this has changed a little bit as the capacity in our region and throughout Georgia has increased over the last several weeks. Really, people that have symptoms can be tested. And we especially should test those that are being admitted into the hospital, especially if they're going into an intensive care unit. People that work in a healthcare facility and have symptoms, first responders, or people with symptoms at work and live in places where there's lots and lots of people and that might include working or living in a long-term healthcare facility. So what will the test tell us? And if you get a positive test, that indicates that there is virus in your airway secretions, and that's an indication that you are infected. A negative test, for the most part, means that you do not have the virus. However, we have to interpret that in light of the symptoms that people have. If they have strong symptoms and they're negative, it's possible they still could be infected and require further testing. And so that brings me up to really talking about the accuracy of this test. And there were some questions about accuracy that were sent in. And so false positives, meaning you get a positive result, but it's wrong, you actually don't have the virus, that's very rare. And usually that would only occur if there was some kind of contamination of the sample or there was some technical problem in the lab. And that just doesn't happen with any frequency. In fact, it's said that there's a 99% specificity of the molecular test. And what that means is if you get a positive test result, we're 99% certain that that is an accurate result. It's a little bit different story for the negative results, and they will occur. And this really comes back to that, what I mentioned a couple slides ago, and that's that we need at least 10 viral genome copies for the test to be able to detect infection. And it may be that we don't get that amount of virus on our swab. And one reason for this, and it comes back to the nasopharyngeal swab, that is an uncomfortable test. Some people don't tolerate it very well at all. And the healthcare provider may not be able to collect an adequate sample through that route, and the test would come back negative. Another factor is that the virus hits multiple parts of the body. We know that. It can get to the upper respiratory tract, which would be your nose and your throat. Lower respiratory tract, it can get into the lungs, and it can also hit other parts of the body, and that includes the digestive tract. And we know that the virus isn't evenly distributed throughout the body in all infections. And so someone that has predominantly tightness of their chest, difficulty breathing, and pneumonia type symptoms may not have sufficient virus in their upper respiratory tract, up in their nose and throat, so that if you do a nasopharyngeal swab, you won't pick up enough virus for a positive test. And perhaps that person should move on and cough up and have a sputum test done to see if they can detect it in the lower respiratory tract. Well, it's important to keep in mind what the test does not tell you. And Fred alluded to this earlier. A positive test tells you that you have viral RNA in your secretions. We take that as a meaning that you have virus, but we don't know simply based on the test if that virus is infectious or not. So we don't know if you're contagious or not. We assume that you are. And if you have a positive test, you're asked to isolate to protect to protect the community and people around you from acquiring the infection. A positive result means that the virus is present, or at least virus RNA is present. It does not tell you your immune status. It does not tell you if you are developing protective immunity or not. And that will be something that Chet will talk about when we start talking about serology. A negative result is a snapshot in time. And so the day that you had that test done, the day they took the nasopharyngeal swab or perhaps sputum, and it was negative, that day you were negative. It does not tell you what your susceptibility to infection will be down the road. And like with the positive test, it just doesn't tell you anything about your immune status, and other testing would need to be done for that. I wanted to talk a little bit about bottlenecks, and many of us have read about bottlenecks or problems with testing in the news. And there are a number of different points where bottlenecks have developed. And I think for many of these, we've had a lot of improvement in our state and in the country. But one of the first bottlenecks is collection sites. So the place where a person could go and actually have a nasopharyngeal swab done. And initially, there weren't very many places to do that. Now, I'm happy to say that those collection sites are increasing. You can have this done at your healthcare provider. There are drive-up collection spots located throughout all of the counties in Georgia. And in some communities, there are ambulatory or mobile testing centers that can go to people's homes and test them. So that's an area that we've been improving. And so bear in mind that the test is collected at one site. So if you went to your physician, most of the time your physician would send that into a lab to be tested. And the capacity of that lab really plays a role in how fast and how much testing can be done. And this has been a bottleneck in the past as well. And it depends on a couple of things. Is one, how many staff do they have to run these tests? It takes skilled workers to run these tests. And some labs have had insufficient numbers to run large numbers of samples. And the second thing is equipment. There's some very specialized equipment that's required to run these molecular tests. And so if a lab has more equipment and more personnel, they have more capacity, they can run these tests faster, and they can turn out more tests in a day. And I think that also is an area where we've seen improvement. Another factor which we still are struggling with is the availability of the reagents and the reagents that are needed to run these tests. And some of these are just the reagents that we need to use with the equipment that will allow us to run these tests. And so sometimes you hear these called kits or PCR kits, and these are sold by different companies, and they are what we need to use with that swab to use in the machine to get a result run. And those have been in shortage. We've had a time where the swabs themselves were in short supply and we didn't have enough to go around. And then we've had shortages in transport medium. So when you take the swab, it goes into a little vial and they add transport medium, and that makes it stable so they can ship it off to the testing lab. And there have been short supplies of the testing medium. And again, I'll give a plug for Dr. Jack Wood's lab. You know, his group in the College of Veterinary Medicine is working on validating reagents and kits, and really a hope of easing some of these bottlenecks and increasing our capacity in Georgia. So we've had some advances as well, and one of them is the saliva test, and that's just going to be a whole lot easier to do than a nasopharyngeal swab. It will take less time, and I think people would be more willing to spit in a cup than they would to have a swab placed all the way back into their deep nasal passages. The limitations of that are very similar to the nasal swab. We still have to get enough virus to have an accurate result. Another advance is what you may have heard as point of care molecular diagnostics. And so this is equipment that's in your healthcare provider's office. And so you can go in, they can take the swab, and they can run it right there in the office. And typically results come back very quickly, usually within the hour. The problem here is that in my understanding, is the instrumentation, the equipment is available, and in many locations, hospitals and healthcare providers' offices, but they don't have the reagents that they need in sufficient supply for this to be a widespread test right now. And so I'll wrap up with this slide, with this last few comments. And in Georgia, I think that the capacity is increasing. And so you can go to the Georgia Department of Public Health, and you can see how many tests are being run every single day, and they update that every day. And it's an impressive number. And so multiple labs are now processing these swabs and running tests. And that includes Georgia State University, Augusta University, Emory, the Georgia Public Health Department, and then private labs as well are running these tests. And as Michelle mentioned early on, the veterinary diagnostic labs and the College of Veterinary Medicine were very interested in assisting in this effort and running COVID samples. And so we'd like to become a testing center along with our sister universities. We're going to need special licensing for that. Right now we're licensed to run animal tests. And so we are actively pursuing getting licensure so we can run human tests. And we really feel that we can assist the testing effort in Georgia. And an example I'll give, and Michelle mentioned this, our Tifton Diagnostic Lab is in a strategic location in South Georgia. And we really think that we can assist in the effort for increased testing in South Georgia. And so, Michelle, I'm happy to take any questions, and that's all I have for right now.
Great, thank you. So we are likely going to run over a bit, but we hope folks can stick with us. We have one last topic to discuss in this particular chat, and that is the antibody test or the serologic test that have been rolled out recently. And Chet Joyner is going to talk to us about that. He is in the Center for Vaccines and Immunology at the College of Vet Med. He specializes in understanding antibody response along with the cells in your body that produce antibodies during infections and vaccinations. He's particularly versed in the body's response to malaria, but has also now switched over to looking at responses in COVID-19 and co-infections of COVID-19 and flu. So I will let Chet take over, and we will also address some of the last few questions that have come in once Chet has given his overview.
Thanks, Michelle. So thank you for asking me to participate today. So as you said, I think a lot about antibodies, and I think a lot about how our bodies respond to infections and fight off infectious microorganisms. And I know that the term antibody, the term antigen, epitope, all these complicated scientific jargon are being thrown around in the news media. And hopefully by talking a little bit about this today, we'll be able to let you know kind of what does this mean and help you make really informed decisions going forward and listen with a critical ear at what people are saying. So I think the first thing we should cover is what is an antibody. So an antibody is a molecule produced by cells of the immune system in response to foreign proteins, foreign substances that ultimately in the context of an infection are meant to neutralize and get rid of that infectious microorganism. So in the case of SARS-CoV-2, your body's going to make these antibodies and hopefully eliminate and neutralize that pathogen and minimize the damage and the disease that it causes you as the host. They're really remarkable little molecules. So down in the left-hand corner, you can see the 3D structure of an antibody. And it's made up of 4 different independent protein chains that are stitched together in the cell before they are released into the extracellular environment in your body, into your bloodstream, etc. And the parts of the antibody that actually interact with the virus are these tips of the ends where kind of the gray and the blue meet and the orange and the red meet on either side. And it's really that part of the antibody that's going to bind to the virus or a viral protein in the case of a diagnostic that can be detected and ultimately hopefully neutralize that virus. This isn't the only way antibodies look. If you look on the right-hand side, there's a ton of different types of antibody classes. So most of what we're hearing about in the media today are IgG and IgM antibodies. As you can see on the right-hand side, they look a little bit different, but ultimately both of these are workhorses of the immune system that really do help neutralize and control a pathogen and ultimately get rid of it. We also have antibodies like IgE. If any of you have allergies, IgE antibodies mediate a type of many different allergies that people in Georgia have against hardwoods, pines, etc. And so these antibodies are made, to protect us against things, but they can also cause us, some annoyances like seasonal allergies, or in the cases of very hypersensitive people, they can also cause allergies and things to that effect. So Michelle, if you go to the next slide. So in addition to kind of looking different and having different functions, these antibodies come up at different points during an infection. And this is a natural part of your immune response of how you generate these different types of antibodies. So let's just pretend that you get infected with SARS-CoV-2, or the virus that causes COVID-19. What's going to happen is that virus is going to begin to multiply in your body, and that is what I'm showing you in the dashed line, up until a point where eventually your immune system goes, hey, wait a minute, this is not supposed to be here. And you're going to start having an immune response. And that immune response is going to lead to many different immune cells becoming activated and starting to try to clear that virus. And one consequence of that is the production of these IgM antibodies. And IgM antibodies are made first, and they're typically only seen against pathogens where, or in new infections. This is something your immune system has not encountered before. And as that begins to increase, you can see that I'm showing you the virus levels start to taper off. So these IgM antibodies kind of hold that virus at bay and keep it contained so it doesn't grow so much that it causes a massive immune response or extensive tissue damage in your lungs or other places in your body. After about 7 to 10 days, you start to see IgG appear. And that IgG goes and grows exponentially, and ultimately, typically in most infections, will clear out the virus and render you immune for a period of time. Now, IgM wanes really rapidly, which is why it's a really good antibody when you're thinking in terms of diagnostics to look for in terms of if you want to see someone who was more recently infected or even maybe actively infected. It's typically gone from your system within about 30 to 60 days, depending on how much an infection induces that IgM response. For IgG, you actually can detect this a lot longer. This stays around many months, in some cases for the rest of your life, like with some of the vaccines that you get. It will stay around for many years, decades even. And you can detect people that had at some point actually encountered this virus or this pathogen in the process. And that's sort of the distinctions of IgM and IgG. And I know that these are terms being thrown around a lot in the media. And I think it's important to kind of think about it in these phases, and that helps you kind of understand what some of these diagnostic tests may tell you. So I know we've gotten questions from the audience, and we've had people are asking lots of questions in terms of what do all these antibody tests mean. And really, one question is, what does a positive antibody test actually tell you? Does it tell you were infected? Does it tell you that you're immune? And if we take that piece by piece, first, your body will make a response to many things, even if it's not infectious. In the case, for example, of pollen against IgE, where we talked about those IgE cells. And so your body, just because you have a positive antibody test, it doesn't necessarily mean you have an infection. It means that you were exposed and probably you were infected. But really, that's at the cutting edge of where we are in understanding this virus, and stuff that groups like mine are really interested in asking about at UGA, along with others here in the Center for Vaccines and Immunology. The next side of that is, if you have a positive antibody test, does it mean you're immune? And the answer is absolutely not. We can make a lot of antibody responses that do not protect us against a virus, a bacteria, a parasite at all. These viruses, we'll talk more about this in the immunity section, but you really have to generate antibodies that can perfectly interact with parts of that virus to prevent it from multiplying and invading their cells. And so just because we have an antibody test that says, oh, you are IgG positive or IgM positive, it does not mean anything about protection. And this is something that many scientists, again, someone like myself, people like Mark Tompkins here in the CVI and the College of Vet Medicine, we're all interested in understanding and asking those types of questions and getting answers for everyone, so that we do know if you have this IgG titer of above 2.5, you actually are highly likely going to be protected, but we really just aren't there yet. I also want to just echo some of the things that other people said in terms of the utility of these tests. What this will really give us an idea of is how many people have been exposed to this virus. And that's a really important thing, because it modifies how we look at it in terms of the number of cases which directly impacts the fatality rate that everyone here is being thrown around a lot. And right now, because, and I know it's getting better, but we have had a shortage of tests, we think that rate is pretty high actually compared to, for example, the flu virus. But actually what we may find is it's more similar to flu. Maybe it is worse than flu, and these antibody tests are essential for achieving that goal and getting a better insight into that. And so I know that there are places in my own county where I live, for example, they're coming to houses and actually screening people for antibodies. So I implore you to please participate in those studies, because they're scientists like all of us going out trying to answer some of these questions that you guys are submitting prior to our webinar. We had a question from the audience about, as a scientist, how would I recommend using these antibody tests for decisions to go to work, attend large group meetings, travel by airplane? And I think that the answer is that right now we don't really know how to use them for those, because we don't have that essential piece of what does it mean to be immune against SARS-CoV-2, and how do antibodies contribute to that immunity? And that's an essential question for us to ask. There is some evidence from animal models and animals that can be infected that you aren't going to have as high a likelihood of being reinfected. However, we must point out that's typically about 30 days. The best data I've seen is about 30 days after your active infection, which is really not a long time, and still well within that period that your IgG could just be going away. So right now, I don't think we have a straight answer on how to use these tests, other than saying that some people were exposed, and maybe they will be more safe after if they re-encounter the virus. In terms of how long are the antibody tests valid, this is an excellent question. We only know really in terms of people when this started and it started being looked at, that these antibodies can persist for probably 6 months. I think historically coronaviruses, some of them that have jumped from animals to humans, you see immunity for about 1 to 2 years, and not immunity, but detectable antibody. And so maybe it will fall in that route, but it's still too early for us to know how long should how often do you have to be tested. And then finally, I think that there was a question about, do we have any tests that we think are going to remain the gold standard? And I think the answer to that is, we've got tests now. They're being developed in many different places, and they all have their pros and cons, very similar to the molecular diagnostic side. And I think that right now we don't know what will be the gold standard in 12 months, but we have a good tool set to continue to improve. And for example, one kind of the holy grail of antibody testing that many of us would love to work on is, how do you develop a test that tells you, have you been exposed, and ultimately are you immune in a fast and efficient manner? And that is kind of, an engineering question along with a basic immunologist question, like for myself and the individuals on this panel.
Thanks, Chet. That was very helpful. So there are a lot of questions that have come in. I think one question I can address: what type of serology testing is currently being done by the Department of Public Health? Both IgM and IgG tests are available, and typically when they take your blood draw, they send it for both of those tests. Another test that came in for you, Chet, is, should we instead be testing for IgA, since it's produced in mucosal surfaces, and could we do it via spitting in a cup?
So I think that it would be interesting to look at IgA, if IgA antibodies are produced and are protective. My feeling is that they may play some role. However, they are a little bit harder to detect and work with than IgM and IgG. And so I think that you could test for them using spit in a cup, for example. But you do have to do a bit of laborious techniques to actually detect it compared to IgG and M. So I think that it is something we could look at, and maybe ultimately to kind of segue into another question we got, maybe we do need IgA antibodies to be produced, for example, from a vaccine to actually get protection. However, we don't really know just yet, and I haven't seen a lot of data on the IgA responses to this virus. It's been pretty focused on IgG thus far, but I'm sure it's coming.
Great. So several questions have come in along this line. One has to do with if it's not clear if natural infection will result in immunity, can you be reinfected? That's one question. I'll open that up to everyone. Chet, in particular, do we know the antibodies that are produced that would provide immunity? I know there are multiple investigators addressing this question. So maybe, Chet, you can tackle the kind of therapeutic antibody question or neutralizing antibody, and then I'll open it up to the reinfection question to everyone else.
Okay, so I think there is some data coming out now showing that natural infections, one out of Emory University where I did my training, and another many other places around the world, that show that you do develop antibodies, particularly they looked at IgG, that will neutralize virus from natural infection. Interestingly, they kind of are acquired a little slower than we would have anticipated, even though they claim it's pretty rapid acquisition. It looks a little slow and slower compared to maybe other vaccines that we've looked at, but nonetheless, there does seem to be relatively rapid acquisition of the neutralizing IgG antibodies. But how do we know if the antibodies produced in the vaccine can provide immunity to reinfection? That's exactly why we're doing vaccine trials. So as I said, you can get an antibody response to many, many different things in response to encountering it. It just doesn't mean that it'll actually provide any protection. And that's exactly the question being asked in these clinical trials that are ongoing with the various different vaccine candidates is, can we actually get antibodies or immunity that is going to provide protection? But I think based on the history of coronaviruses, that it is likely that can probably occur. But it may take a few tries to be successful.
And so anybody have any comments about reinfection? We heard early on in some literature that hadn't been peer-reviewed that at least in South Korea, folks were testing positive on the molecular test, and then 30 to 40 days later, testing positive again on the molecular test. I think information has come out since that this was just viral fragments still in the body from the original infection. Do we have any evidence or data yet about whether or not people can be quickly reinfected after clearing the virus?
Can I take a shot at that? So the way I would potentially answer that is looking at history of other respiratory diseases, particularly when you're in an endemic or during the open season for the particular disease. There's absolutely no reason, and again, this goes to the question of the type of test that was used, and in this case, it was the PCR test to look at the presence of the virus in the nasal swab. And the issue with that is colonization, again, or carriage state or asymptomatic carriage. You can be recolonized with any organism multiple times. And again, giving a different example, when the pneumococcal vaccine was first introduced, it was demonstrated that individuals could be carrying Streptococcus pneumoniae bacterium in their upper airways for weeks or months at a time. Then it would lose it, they would gain it back, and then they would lose it again. So without any symptomology whatsoever. So the fact that you've got the organism in your respiratory tract does not indicate that you are actually infected. It just means you are being exposed to someone who is generating the organism. And so it's hard, you can't put those two pieces together at this point with the lack of diagnostics.
Great. Okay. One other question that came up is, do we know the latency period of this virus? So do we know if you're exposed to it, does it sit dormant while you're shedding it? And if we don't know this, I'm sure you all have ideas of folks in the college that are actually addressing this question. I'll open that up to the panel as well.
So, Michelle, this is Mark. I'll take a shot at it. So, one of the things that I guess astounds me a little bit is why people are not asking the question, what happened with this SARS-CoV-2 virus, where it got into the population and continued to spread, and we're not able to control it. Whereas before the original SARS virus, we were able to eradicate that fairly quickly, relatively quickly, if you will. And a lot of it has to do with the incubation period, or you said latency period. This really doesn't go through a latency, but it's the incubation period of the virus once a person is infected, until they start showing signs of disease. That period for the original SARS virus was very short, just a matter of a day or two, to where they started showing signs of disease, so our contact tracing could be very specific for those particular individuals. This virus, the COVID-2 virus, the period of time from when a person becomes infected and when they could start showing signs of the disease or symptoms, that can be as long as 14 days. And during that period of time, there's enough virus there in that individual that they are transmitting it. So someone can walk into the grocery store looking perfectly healthy and shedding quite a lot of virus and infecting other people around them. That's what makes this virus so difficult to control, is that period of time where they're not showing any symptoms, but they are highly infectious.
Great, thank you. One last question before we wrap up. This is probably in Chet's wheelhouse. So the timeline for widespread antibody testing and whether or not there appears to be a bottleneck. Chet, you want to handle this one?
Sure. I think that we're getting to where there can be widespread antibody testing. So I'm sure many of you know that, Quest Diagnostics and other places do have antibody tests available. The word of caution to that is many of these tests have kind of been released under that what we call the emergency use authorization with the FDA. And what that essentially, without being too complicated, means that it hasn't gone through the same type of rigorous review that most tests, for example, your cholesterol test or your glucose test, et cetera, would go through before being put onto market. And what that means is that it facilitates the widespread antibody testing that everyone really wants to happen. And we really do need that to happen to get a better hold on how many people have been exposed potentially to this virus. However, it doesn't mean that they're all equal. And so I think that the time we're in a phase now, based on how the FDA is responding, to where they're now starting to really rigorously start re-reviewing some of these antibody tests, and probably the ones that are the most accurate in terms of predicting having had an infection from actually using samples from patients that had the infection, as well as people that may have been known to not been exposed to the infection to the best of our ability, will start to guide us as to what's the most appropriate test to fund and actually start viewing at the population at large. The bottleneck to all antibody testing is that it actually it can be automated and things, so that is a good thing. But it really is kind of the sample collection and the laborious nature of doing these assays. There are tests that are out there that are quite rapid. Some, for example, in malaria, we have ones in endemic when we're in the clinic in Africa that we can actually just prick the finger, similar to what was shown on the diagnostic slide, and have like a quick little readout that tells you 2 lines means positive and 1 line means negative. I think we're moving in that direction, and that's really going to help accelerate this testing process. But right now, I think kind of one of our bottlenecks is, one, having the antibody test readily available to everyone that wants to get tested without, a price tag attached to it. And then two, getting it into a format where it really is a rapid diagnostic test. And I know, for example, that Dr. Tompkins here in the CVI is working with the Georgia emergency unit to develop some small deployable kits where you can actually do these antibody tests effectively in the field. And this is an important area for people to continue to think about and work towards.
Great. So just to wrap up, I know we went a little bit over. I apologize for that, but I think we did pretty good considering you have like 5 scientists on the phone. So I just wanted to kind of sum up some of the areas, at least in diagnostics, that your UGA scientists are working on. In particular, the veterinary diagnostic labs are trying to get up to speed, and also licensing to start running the molecular diagnostic tests, particularly in South Georgia. As you just heard from Chet and others, there are faculty working to develop rapid point-of-care tests that don't necessarily require some of the expensive machinery. So this is akin to tests that would be run in third world countries, and just allow folks easy access, more capacity for the testing. Again, to plug Mark Jackwood's group, testing of new kit reagents and tests themselves, validating them so that we can address the bottlenecks in testing as we need to ramp up with states in the country opening back up. And then also antibody diagnostic kits to identify those antibodies that are protective and how long those protective antibodies actually stick around. And so it's this last bullet point that will lead us into our second chat, which will be at the same bat time and same bat channel next week at 1 p.m. And here we're going to actually dive into what is driving the immune responses in various people and children when they're seeing this virus for the first time, and what do we know about why some folks are ending up on ventilators and other folks don't seem to have much of a respiratory symptoms at all, but maybe like in week 3 are coming in with blood clots. So kind of the spectrum of how this disease is progressing through the human population. And so Chet will be back with us, and we'll also have Kim Klinowski, Karen Norris, and Blazis Rada. And so we hope to see you all next week, and stay safe and wash your hands. And thanks again for joining us.